Good morning, afternoon, and evening. It's Wednesday, April 15th, 2020, and on behalf of everyone at Simulations Plus, welcome to DDI Predictions with GastroPlus with today's presenter, John DiBella. For optimal video and audio connection, we highly recommend you close any additional web or streaming applications that may affect your internet bandwidth. This call is being recorded. Today's course structure will have lecture and examples followed by a live Q&A session. So grab your coffee, tea, or water. It's going to be an insightful 90 minutes. Now, before we get started, there's a quick poll question. How would you classify your experience with GastroPlus? We'll give you about 30 seconds to submit your answer. Few more seconds left. And we're going to close the poll now. Thank you for providing your answers. We've got lots of beginners and intermediates, so you are in the right place for today's presentation. Now it's my pleasure to introduce the moderator, Mr. Dan O'Connor, Director of Business Development for Simulation Sciences. Take it away, Dan.
Thank you very much, Arlene, and welcome to the third day for our workshop from home seminar series for GastroPlus. We're looking forward to a very energetic interaction with you today, and thought I would start by again inviting, for those of you that are spending the third day with us, you may have seen this slide before, but we do want to invite your interest and participation if we can help in any way with respect to defeating COVID. If you have a research program that's underway, a clinical trial, or we can be at all helpful. We remain very much interested in being able to assist you in that effort. Thought I would just share a quick slide that summarizes the contribution of GastroPlus to a variety of areas worldwide. We've had over 35, I think it's fast approaching 50, approved drug product applications that have been supported by specific GastroPlus simulations. Obviously, companies and regulatory agencies worldwide are using the tool. And if there were ever a time in the history of drug development where modeling and simulations were center stage, it couldn't be better thought of than at this time in history. It's my pleasure this morning to introduce the presenter, who's John DiBella. John did his undergraduate and graduate work in biomedical engineering at Case Western Reserve University in Cleveland, Ohio. John has spent the entirety of his professional career, what will soon be upwards of 17 years, working with Simulations Plus in Lancaster, California. He started as a modeling and simulation scientist, and after a couple of years of significant contribution to the science, he became a senior application scientist. Then he moved to sales and marketing arena and was promoted to the position of Vice President of Marketing and Sales. Just about 3 years ago, John was named president of the Lancaster division of Simulations Plus, that being the division that authors and promotes GastroPlus. John, in my estimation, is one of those rare individuals who's enjoyed great business success while at the same time maintaining his world-class scientific acumen. John is a modeling and simulations expert. So it's my pleasure to introduce John DiBella.
Dan, thank you very much. Your check is in the mail. And hello, everyone. Welcome to day 3 of the Workshop from Home program. For those of you who are attending for the first time this week, you've chosen a very good topic, a topic that we are very much interested in promoting more use of GastroPlus towards. For those of you who did attend either or both days 1 and 2, welcome back. Hopefully you've had pleasant dreams of GastroPlus these past couple of nights, and I hope to meet the expectations set by Maxime and Vieira the last 2 days. What I'd like to do today is take you through some theory with drug-drug interaction predictions, but do my best to spend the majority of the time that we have together today going through some hands-on applications. We're not reinventing the wheel when it comes to the underlying equations and the calculations of the changes in AUC ratios, whether we're using static steady state approaches, whether we're using dynamic mechanistic approaches. So what I would like for you to walk away with today is a feeling of comfort in being able to take whatever data you have at various stages of your project, whether it's early on, looking to try and assess metabolism-based DDI liabilities. And as we start to then generate and capture more and more data, refining our models, doing more mechanistic predictions to ultimately inform regulatory decision-making. Now, I would like to just expand on a couple of the statistics that Dan had shown on one of his slides. The first being the number of approved drug product applications that have been supported by GastroPlus simulations. As Dan said, the conservative number that we are aware of today is over 35. In reality, this number is likely much higher than that. And there is great diversity in the types of applications for which GastroPlus results have been applied. You can see here a short list of some of these approved drug products, and what some people may not be aware of today is that there are real examples of being able to use GastroPlus DDI predictions to support applications in the spaces of metabolism-based DDIs or transporter-based DDIs or absorption-related DDIs. Besides those drug-drug interaction predictions, there are also opportunities, of course, to utilize the physiologically based biopharmaceutics modeling approaches to help establish specifications around the overall products or critical material or property attributes. So we feel comfortable and confident in saying that there is really no other PBBM, PBPK platform that has the diversity in applications for supporting regulatory interactions. I also just want to expound a little bit on the over 1,000 peer-reviewed journal publications and conference presentations. While we've been very fortunate over the years to have many of our clients presenting and publishing work in the spaces of absorption modeling, helping with formulation assessment or optimization work, helping with food effect predictions, helping with population predictions, BE trial simulations, and so on. I also want to make the audience aware today that there is a lot of literature highlighting the use of GastroPlus to support drug-drug interaction predictions. And there's been a lot that has been published just over the last couple of months as we entered into 2020. So please, after the webinar today, as you absorb some of the lessons that we learn, take an opportunity to go through some of these publications and learn how your peers are applying their GastroPlus models for drug-drug interactions. What we're going to be talking about today is two types of interactions. The first being dynamic, where we can begin to apply the PBBM or PBPK models that we have optimized and validated utilizing a combination of preclinical and clinical data, and run dynamic DDI simulations where we can account for a variety of different mechanisms, be it competitive or time-dependent inhibition and or induction. And with these dynamic predictions, we can account for metabolism and or transporter-based interactions. We'll also, though, spend a little bit of time talking about the static or steady state options which are available within GastroPlus, because there's a real opportunity to leverage early in silico machine learning predictions with in vitro data to do some of that DDI liability assessment work. And with the static steady state approaches, we can account for competitive and time-dependent, also induction, but we are going to be focusing exclusively on metabolism-based interactions. As we're going through and discussing static approaches, mechanistic or dynamic approaches, please always keep in the back of your mind this idea of taking some of the lessons that have been learned, especially in the introductory course, where we're going to always try and do the best we can in building up a bottoms-up type model. We want to be able to take all of the effort that has been invested and leverage all of the data that has been collected to build up our PBBM, PBPK models, and not minimize the impact of the site of administration, the local exposure aspects, whether we're talking about oral absorption, whether we're talking about non-oral delivery routes. Everything that we're doing to build up the predictions of dissolution and absorption at the site of administration will down the line be impacting our estimates of concentrations in liver, in kidney, in other tissues. And so having that correct PBBM model in place is going to be critical for generating with confidence correct DDI predictions. Now, one of the ways in which we help at Simulations Plus and with the GastroPlus platform in particular is by providing you with a DDI standards database. This table here shows the list of currently available validated PBPK models that we provide in this database. And you'll see there are now approximately 40 drug models which are provided, covering many of the necessary CYP and UGT probe substrates, inhibitors, and inducers. And we've been making a real effort over the course of the last 12 to 18 months to also build up our database to account more for transporter-based interactions as well, including some common transporter inhibitors, probe substrates, and or inducers. You see that in parentheses next to each of these compound names a year, and this is an indication as to when these models were last built and updated. And one of the efforts that we have ongoing is to review literature and update models and add in new mechanisms when possible. And as we're doing this, we're going to be compiling our word reports, our documentation showing how these models were developed, how they were validated and verified, so that you can very easily run your simulations with your NCEs using any of these compounds. And as you're preparing your reports for submission, you'll be able to include the supplemental documentation showing and describing how these standard models were built, validated, and verified. And I also just want to point out that in the formal standards database, we provide approximately 40 drugs. But of course, over the 20 plus years that Simulations Plus has been in existence, we've built a lot of different models. And so the non-substrate, non-inhibitor, non-inducer compound model files that we have available would easily be in the hundreds. But those that are specific to drug-drug interaction predictions are included with the DDI module in GastroPlus. So let's spend a couple of minutes just briefly going through some of the theory, first talking about the static or steady state predictions. And again, I want to emphasize that we're not necessarily reinventing the wheel here. When we're talking about the static or steady state approaches, what we're going to be trying to do is simply calculate AUC ratio changes, accounting for any contribution of the gut towards the DDI, towards changes in AUC, multiplied by any contributions from the liver towards that DDI calculation. And when we're talking about our victim compound, what we're going to be focusing on is really just providing estimates of FG, the fraction that would be escaping gut metabolism, and the FM percentage. What is the fraction of the compound being metabolized by any of the CYPs or other enzyme systems? That's it. We're not interested in doses. We're not interested in concentrations for the victim compounds. All we're interested in at this stage is FG estimates and the fractions metabolized. Then, depending upon the perpetrators that we select, we would need to have some information about inhibition parameters and or induction parameters, and also have some estimates on the concentrations in the gut and or liver. The equations can be extended for multiple perpetrators. If we have multiple competitive inhibitors, then this term in the equation would be expanded to cover those multiple perpetrators. If we have multiple time-dependent inhibitors in our calculations, this term would be the one that is expanded, where we would have for each of those inactivation rate constants, degradation rate constants, along with the inhibition parameters. And then for multiple inducers, it would be this term that is expanded, having the unique Emax, EC50 values along with concentration estimates. So that's it. Again, static steady state methods. What we're doing here is just calculating using these algebraic relationships what would be the AUC ratio changes in the presence of the perpetrators. So the required inputs, fractions metabolized and FG values for the substrate for our victim drug. And then depending upon the mechanism, the KI or IC50 for the inhibitor, the inactivation rate constant for the inhibitor if we're working with a time-dependent inhibition process. If it's induction, then having EC50 and or Emax values. Also, if we're talking about TDIs, having some estimate of the degradation rate or turnover rate for the enzyme will be important. So having this information for the perpetrator plus generating an estimate of that steady state concentration for the perpetrator. And let's spend a minute or two talking about how the various ways in which we can calculate that concentration. We'll talk more about setting up the types of interactions when we go into some of our first examples coming up. But with regards to the concentrations, there are going to be several options for calculating what we would call the effective or static or steady state perpetrator concentration. The first option, the calculated concentrations, are just obtained from standard equations. Again, not reinventing the wheel. All we're doing is utilizing the definitions from the ITO publications, the definitions that have been outlined in some of the reference or guidance documents, and we use these algebraic relationships to calculate either systemic, average, minimum, or maximum concentrations. If we're using in vivo parameters, if we're working with in vitro inputs for the inhibition or induction parameters, we have some equations for calculating liver and or gut concentrations. That's it. Now, where we are able to incorporate mechanistic type approaches into these static or steady state methods is by simulating the perpetrator concentrations using those compartmental or PBPK models available for the different standards in our GastroPlus DDI database. So if we decide that we want to leverage those validated PBPK models in place, we can simulate what those concentrations would be. And depending upon the dosing regimen that we set up, if we run our simulations and the perpetrator reaches steady state before our victim or substrate compound is administered, and we see in this plot here, that would be this red vertical line, then the plasma or liver effective concentration is going to be estimated as just a simple average of the simulated peak and trough concentrations. If we have not reached steady state before our substrate or victim drug is administered, again represented in these by these red vertical lines here, then what we would do is calculate the effective concentrations using an AUC-driven average concentration across that dosing interval from the time of the substrate administration to the next dose. So here we would then be using an AUC-driven method to calculate that single point average concentration that would then feed back into the AUC ratio changes, the DDI calculation. With regards to the gut, if we want to include the contribution of the gut towards the DDI assessment, then the effective concentration in the gut is going to be estimated as the average enterocyte concentration across all the compartments. And so here you see an example slide where the substrate is administered, and we can run these simulations to track what would be these enterocyte concentrations in the different compartments, in this case of the ACAT model. And then we take that average enterocyte concentration across all these compartments to have our gut concentration input for the perpetrator. That's it. We'll talk a little bit more about the concentration selection window when we go into one of our first examples, but we will have options for utilizing either total or unbound calculated concentrations, which again are just those simple algebraic equations, or leveraging the simulated concentrations, total or unbound, and calculating from the simulations the steady state concentration estimate, depending on what we saw in the previous couple of slides. So we just spent a little bit of time talking about how we get those concentration estimates for the perpetrator. Now let me just spend a minute or two talking about ways in which we can start estimating values of the fractions metabolized from in vitro data. We will go through a first case study coming up where we'll start with our machine learning predictions to inform the DDI assessment, but then we'll work with some in vitro data collected from either recombinant CYP assays or liver microsomes, and have the very nice window and tool in the program that will allow us to load in our in vitro data. If in the case of working with recombinant CYP data, we would estimate or define our intersystem extrapolation factor. And from that, calculate what would be the intrinsic clearance for the individual CYPs, and take that intrinsic clearance for the individual CYPs, dividing by the total CYP clearance to help us estimate our fraction metabolized. So if we're working with recombinant CYP in vitro data to help us capture the FM percentages, then we would have to have the recombinant CYP clearance measurement for the individual enzymes, along with our standard intrinsic clearance measurement in human liver microsomes to capture that ICEF value. And from there, the program will help us estimate FM percentage. If we're working instead with liver microsomal data, then what we'll be doing is utilizing our intrinsic clearance data measured in liver microsomes and multiply that by the intrinsic clearance in that microsomal assay, which was also co-administered with a known inhibitor for a specific CYP enzyme, to give us our intrinsic clearance for that particular isoform. And we'll be using some additional scale factors accounting for the differences in expression levels in vivo versus our in vitro systems. And then using that intrinsic clearance for the individual isoform, dividing by the total, correcting for any estimates of non-CYP clearance that we define, we'll have our FM percentages to feed into the DDI calculations. With regards to validating some of the underlying equations, not only the equations that I showed earlier, but also some of the inputs that we provide for those standard substrates and or inhibitors in our DDI database. We've been presenting and publishing work now for well over 10 years. And here's just one example of work that was presented at the AAPS meeting in 2010. Michael Bolger, our chief scientist, was first author, and this was work that was done with colleagues from Simulations Plus and also in collaboration with Roche. They have been very good partners of ours for many years and were instrumental in helping us design and validate the DDI module in GastroPlus. And what we see in this table, let me just zoom in here briefly. We evaluated a number of different published DDI studies for different enzyme systems and compared our simulated changes in AUC ratios versus what was reported and observed at that time. And you can see in the poster that for the 20+ studies that were evaluated across the major CYP isoforms, utilizing our built-in substrate and or perpetrator models, we did a very nice job of being able to capture the observed AUC ratio changes relative to what we predicted. So this is some of the very first work that we presented on, highlighting some of the validation of the underlying DDI equations, the DDI model inputs for a variety of different CYP isoforms. And this work is available to download on our resource center at our website, www.simulations-plus.com. So if you're interested in a copy of this work to go into more detail, it is available for download. So I've set the stage for some static or steady state DDI predictions that we can perform very early on, leveraging whatever limited in vitro, or where we're going to start with this case study, machine learning inputs to predict and assess the DDI liability. So we're going to be going through here now an example with risperidone, one of the compounds that we worked with on day one, and talk about how we can start to inform our DDI predictions at a very early stage with chemical structure, and then leverage any additional in vitro data we might collect to refine some of our inputs. Before we go through this case study, I believe we might have a poll question. Is that right, Arlene?
Yes, thanks, John. Here we go. Do you currently have access to the DDI module in GastroPlus? We'll go ahead and give you a couple of seconds to submit your answer. It's okay if you don't know or you click no. We can help figure out how to get you access if you have a current license, or an evaluation if you need to get access. So closing in on a couple seconds here. All right, so it looks like 53% of you don't have access to the DDI module. So stay tuned to hear more from John now.
Thank you very much, Arlene. And I'm sure for those 53% who do not have access, many of you will be asking for it to management after this training. So let's go through now this example here, and allow me to slightly go off the rails a little bit and take you through briefly the ADMET predictor platform, which is where we'll be generating some of our SIP-based metabolism predictions, estimating things like sites of metabolism, predicting metabolite structures that are likely to be formed, and for the major SIP isoforms, also being able to predict intrinsic clearance values as well. So if I exit out of the slides for a second, what I'm going to do, this is the standalone ADMET predictor platform. I know many of you today leverage the machine learning models that we develop in ADMET predictor through the plugin or module that is available in GastroPlus. And that's great. That plugin does an excellent job of providing the inputs necessary to set up an in silico PBPK model, which is something that Maxime discussed on day one of this workshop from home series. What I'd like to do is just open up this risperidone mol file, the chemical structure for risperidone that we sketched in our MedChem Designer software. And when that structure is loaded, we then have an opportunity to be able to trigger a variety of different property predictions through the toolbar here at the top, or these option buttons down at the bottom. What I'm going to do is click on this blue Add Me button and calculate the set of physical, chemical, metabolism, and toxicity endpoints for this compound. And we'll see that we've calculated in a fraction of a second over 140 different ADME tox endpoints, each of those endpoints being represented as a different column or cell in this spreadsheet. Now, I'm not going to go through in any great detail all of these individual property models, but I will go over towards the section where we start to generate some predictions for metabolism. So the machine learning models that we have today predict for 9 different SIP isoforms whether or not your molecule is likely to be a substrate or non-substrate. If it is predicted to be a substrate, then it will attempt to identify the metabolic hotspots on the compound. And then for the 5 major CYP isoforms, 1A2, 2C9, 2C19, 2D6, and 3A4, it will predict the metabolite structures that are likely to be formed and also predict the intrinsic clearance rates. So I see here for risperidone as an example that it is predicted to be a substrate for CYP1A2. And if I double click on this CYP1A2 site cell, I can open up this window here, which shows me these red circles on the molecule indicating the metabolic hotspots that are predicted for the 1A2 enzyme. And on this window, I can select all of the different sites of metabolism models. So here are the 9 CYP models that we have. And I'll just blow this up. And wherever I see here red circles on the molecule, that indicates that for this particular isoform, this compound was predicted to be a substrate. And the red circles indicate the potential metabolic hotspots. So this gives me some very early clues as I start to prepare for downstream experiments to run, which isoforms I may want to focus on first. Out of all the various isoforms I could potentially test, I can leverage these machine learning predictions to help me inform or prioritize the experiments going forward. And so I see here that it is predicted to be a substrate for CYP1A2, predicted to be a substrate for CYP2D6, and also predicted to be a substrate for CYP3A4. If I double click on the structure here, I'll open up the MedChem Designer tool that we offer. And this is our free, yes, I said free, sketching software that you can download from our website, which allows you to draw up to, I believe, 100 compounds in the canvas window using the controls or buttons on the left side here. With the free version of MedChem Designer, you're able to save your molecules that you've sketched as mol files, SMILE strings, RDF, SDF files. And you're also able to leverage our prediction models for log P, log D at 7.4, polar surface area, number of hydrogen bond donors, number of hydrogen bond acceptors, and Lipinski's rule of 5 calculation. If you have access to the ADMET Predictor program or the ADMET Predictor module in GastroPlus, what you're also able to do with the MedChem Designer tool is predict metabolites that are likely to be formed. And so if I have my structure here, I can click on this CYP or this MET button in pink, which will allow me to predict the metabolite structures from one or more enzyme systems. And so here, within a fraction of a second, we have now generated our predictions of the metabolites that are likely to be formed by the CYP isoforms for which our parent structure was predicted to be a substrate. You'll also see these percentages next to these metabolite labels, indicating essentially what you can think of as a yield. So if this is the total intrinsic clearance that is predicted for the parent risperidone compound, you can see that this M2 metabolite has a yield of 54%, M3 at 24%, and M5 at 17%. So this starts to give you some clues about which are the likely metabolites that will be present if we were to do any further in vitro med-ID work, or we start thinking about taking this structure into some PK studies. And I just want to end with this brief demo of ADMET Predictor by highlighting the parent structure again. And you can also call models that we have for non-CYP systems to predict any glucuronides that may form. In this case, risperidone is predicted to be a non-substrate for the UGT enzymes, and also any potential esterase metabolites that may be formed as well. But again, predicted to be a non-substrate for the esterase systems. What I'd like to do now is take some of these predictions of metabolism rates, of intrinsic clearance rates, and use those inside of GastroPlus in the DDI module to help me calculate FM percentages for risperidone and do some early DDI liability calculations. So now if I transition over, let me just minimize ADMET Predictor and go to GastroPlus. What I'm going to do here to start is create a new database project. We'll give this project a name. I'll call it Drug R because I barely remember how to spell risperidone. And once I have that new project created, what I'm going to do is now import that same structure file that I imported into the standalone ADMET Predictor platform. So when I call the module inside of GastroPlus, I do that by going to File, Import, and this Import Structure menu here. And I'm just going to go to my desktop and open up that same structure file. Within a fraction of a second, the program processes the machine learning models that would be important for informing the GastroPlus simulations. I have this import structure properties window that pops up. And what I can do here is, in this window, start to set up my in silico PBBM PBPK model. I'm going to be using here just all in silico inputs for the purposes of the exercise. But I do want to create a new PBPK model. From the exercises that Maxime went through on day 1 with risperidone, what he had done is created a Japanese population group. And I believe he assumed it was a male subject, considered healthy, and will create the age as 35 years old. So it's very, very easy to be able to create PBPK models for different population groups, health status, age ranges, and so on. I'll just click OK here to create that file. And what we're going to be doing is just utilizing our predicted in silico intrinsic clearances from the machine learning models, those models that were developed with recombinant CYP data as the underlying training set. And I'll click OK here. When we import a structure file into GastroPlus, the program is going to be adding a new record to the end of our project file. So here is the risperidone structure. We have all of our in silico inputs for the physicochemical properties. We have the baseline GI physiology that is set up, in this case under fasted conditions. And on the pharmacokinetics window, we have our 35-year-old Japanese male healthy subject. We've got our PBPK summary grid here that shows us our predictions of the partition coefficients for the various tissues in the PBPK model. Summing these up and multiplying by the respective tissue volumes gives us our estimate of the volume of distribution. So this is the mechanistic VSS that is calculated from the Lukachova approach. And then you can see here that we have our estimates of the total clearance in liver based upon our ADMET predictor machine learning model. And we also have a small fraction of passive filtration clearance in the kidney that has been calculated using the fraction unbound in plasma and multiplying by the GFR for this 35-year-old Japanese male subject. And summing up the clearances here gives us our total in silico systemic clearance of around 26 liters per hour, and ultimately our half-life of around 6 hours. So this is all fine and dandy. And I know Maxime on day one took us through this setup here. What I'm interested in, though, is not so much the simulations that we could run in this main GastroPlus window, but I want to go directly into the DDI module and do this DDI liability assessment. If I go to the modules menu and go down here to menu item number 8, DDI, this is where we can now call and trigger the DDI module in GastroPlus. Now, when you're seeing this module for the first time, I remember what it was like to learn it, so I can be empathetic. Once you have used this once or twice, the setup of this window becomes very logical. The left side of the DDI module window is going to reflect any inputs that have been set up for the compound, for the current record on the main GastroPlus window. So here is this second record, risperidone, which is the in silico record that I've set up. And the left side here now reflects my compound by default being treated as the victim. And I have populated here the various intrinsic clearances that were calculated through the major CYP isoform pathways and their respective FM percentages, all populated for me right now. If my compound, if the current record, and we'll see this coming up in one of the next examples, if my compound is actually a perpetrator as well, then I would just make this switch here and start filling in any perpetrator information that I might have. But at this early point right now, what I'm concerned with is my compound has been identified as a substrate for 1A2, 2D6, and 3A4. And I want to know if it's being co-administered with known inhibitors or inducers of those enzymes, what sort of DDI liability would I have? So if I go back here and treat my compound, this current record, as the victim, on the right frame here of the DDI window is going to be where we select the interacting compound. And by default, the program is going to load the DDI standards database. Also by default, the program is going to filter the database and only show those perpetrators in our database which are known inhibitors or inducers for one or more of the enzymes that have been defined on the left side here. So when I click on this drop-down menu, you can see I don't have 40 odd records in this drop-down menu because the program has automatically filtered out all of those compound model files where we do not have any inhibition or induction parameters in place for 1A2, 2D6, or 3A4. What I'm going to do is I want to select from this list here fluvoxamine, PBPK. And the reason why I'm choosing fluvoxamine is because it is a known inhibitor for 3A4, 2D6, 2C19, and 1A2. And so if I want to see what would be the impact of dosing my compound against a molecule which is an inhibitor for one or more of these enzymes, fluvoxamine serves as a good case study. So what I need to do is, once I select my interacting compound, I can then decide which of the inhibition or induction parameters I want to use for the calculations. So you can see here in our standard model for fluvoxamine, we only have one reversible in vitro total Ki from literature, which is reported as 10 micromolar. And as I scroll across this table here, the last column is going to show me the reference from where this data has come from. So I want to decide here to use this reversible in vitro total Ki for 3A4. For 2D6, you can see that I have a reversible in vitro total Ki and a reversible in vitro unbound Ki as part of the database. It's going to be important that when we start to set up the inputs for our DDI predictions, that we are consistent in terms of using in vitro inputs only, in vivo inputs only, total values or unbound values. And if there is a mix, we need to make sure that we are correcting so that the program can consistently work with all total, all unbound, all in vitro or all in vivo. So in this case here, for 2D6, I want to use, since we only have for 3A4 the total in vitro value, I'll use the same here, this total in vitro reversible Ki for 2D6. 2C19 is not important for me right now because my compound was predicted to be a non-substrate. And for 1A2, you can see that we have in vitro and in vivo total and unbound Ki's that are reported from literature. And what I want to do here is I'll select to, again, be consistent with my selections for 3A4 and 2D6 to use the in vitro total Ki value. So for the perpetrator, I need to select my inhibition or induction parameters, and I've done that here. I would need to set up my dosing information, and fluvoxamine is typically dosed at 100 milligrams, and we're assuming a once-a-day dosing. For the compounds that are in our standards database, we do provide the information that would be needed to use the calculated concentrations that we saw in some of the earlier slides, though you are free to come in and modify any of these inputs if you'd like. And then I have to make a decision here to select which steady state concentrations I want to use as the input into my predictions. So for fluvoxamine, do I want to utilize systemic total concentrations or unbound concentrations? Do I want to use liver or gut total or unbound concentrations, assuming the algebraic relationships, the calculated approaches? Do I want to use simulated concentrations to generate those estimates? Again, total or unbound. Or do I have any user-defined concentrations? Do I just want to type in a concentration value to use? Now you'll notice straight away with the default selections for the concentrations, there is a red label down here in the lower right corner, which says there is a Ki concentration mismatch. Click here for more information. If I click on that label, it will open up a very useful help window that will pop up, which gives you some clues which concentrations you should be selecting based upon the inhibition or induction parameters you've chosen for the perpetrator. So for steady state or static predictions, if you're using in vitro unbound inhibition or induction parameters, you should be using unbound liver concentrations. If you're using in vitro total inhibition or induction parameters, you should be using total liver concentrations. If I'm using in vivo unbound or total inhibition or induction parameters, then I should be using unbound or total plasma concentrations as the input for the DDI calculation. So, and then you see this note here that in dynamic simulations, when we go to the next example, the unbound concentrations at the site of metabolism or for carrier-mediated transport are being used. So the most relevant inhibition or induction constants would be, if possible, in vitro unbound Ki, IC50, or EC50 values. So if I go back to the DDI window, the reason why I have this mismatch here is because I've got in vitro values selected that are total. And I've got one of my selections here being plasma or systemic concentrations. That's a mismatch. I should only be using liver concentrations here. If I click OK, I still have the mismatch label because I have selected here the unbound liver concentrations, but I'm using total in vitro inhibition parameters. So I should be using total liver concentrations to match up with the total in vitro inhibition parameters. And if I click OK here, that label goes away and I see straight away the AUC ratio calculation for the liver, slightly above 1. The total AUC change, slightly above 1. But according to the FDA guidance, any AUC changes which are less than 2 would indicate a weak or no interaction. Let me just go back to the slides here quickly. So what we've done here, right now we don't have any contribution from the gut to the DDI because the machine learning predictions that we set up here have only placed these enzymes into the liver. So this ratio here stays at 1. And what we've done now is we have used the fraction metabolized by the different enzymes. We've used the in vitro Ki values. And we've used, in this case now, the calculated liver inlet concentration here as the input here. And that then gives us an AUC ratio change of 1.14. Now, this is fantastic. I've been able to do this very quick DDI liability assessment using just my machine learning predictions here. But let's say I have applied these estimates to help guide some of the in vitro testing that I want to do. So if I go to the activity slide here, I've got some microsomal clearance data that was measured in the presence of known inhibitors for 1A2, 2D6, and 3A4. So I have my total intrinsic clearance in the microsomal assay of 269. Then I have what that intrinsic clearance would be in the presence of the inhibition of the individual enzymes, along with the abundance in my microsomal assay for those different CYPs. So what I'm going to do now is go back into the DDI window. On the left side here, back from my compound, there is a button that says calculate FM values. And if I click on that button, it's going to open up this very convenient window, which will allow me to select the population type, which we'll say is Japanese, the percentage of our non-SIP clearance, in this case, we'll say is pretty low. We'll call it only about 5%. That's based upon the FUP GFR value. The total uninhibited intrinsic clearance from our experiment in vitro was 269 microliters per minute per milligram protein. And we're going to just enter in here the values for 1A2, 2D6, and 3A4 inhibition in those assays. So the intrinsic clearance that was measured for 1A2 was 240 when it was inhibited. And the content level was 42 picomoles per milligram protein. For 2D6, the intrinsic clearance measured in the presence of 2D6 inhibition was 200, with the abundance level set at 9.1 from the experiment. And then for 3A4, the inhibited intrinsic clearance is 100. And the abundance level is 7.8. Oh, excuse me, 78. Sorry. So now I have, from the in vitro data, these FM percentages. And what I can do is I can export these values back into the main DDI window. So now I have 5.22, 34.9, and 54.87 transferred back into my main DDI window. And I can close out of here now. And so what I can do is come back in here and now select, excuse me, what would be the inhibition parameters to use for 1A2, 2D6, and 3A4. And you can see now, using this in vitro data, there's a little bit of a change, but not substantial. So this is a way in which we can start to do some very early DDI liability assessment when we take our machine learning predictions and or in vitro experimental data and select an interacting compound to see whether or not there are likely to be any weak, moderate, or strong interactions in place. Now, if we go to that next step, this compound or another compound is being progressed through development. We're starting to generate a lot of PK data. We start to build up our baseline PBBM or PBPK models. And we want to now do the more formal mechanistic or dynamic simulations. Let's just talk briefly about some theory here and then go through one quick example. So for dynamic DDI simulations, what we need to do, the heavy lifting here, is going to be really on developing the baseline models, leveraging all of the PK data from preclinical or clinical studies, building up and validating that baseline model. And once we have that model in place, then the DDI predictions are relatively straightforward, at least the setup. With dynamic DDI simulations, we're only going to be limited by really our imagination and our ability to parameterize. Dynamic DDI simulations will account for interactions in any tissue, account for competition between multiple substrates for the same enzyme or transporter, competition between multiple irreversible inhibitors for binding to the enzyme, accounts for the possibility of perpetrators acting as inhibitors and inducers, and any enzyme or transporter interaction can be modeled here. Remember, with the static steady state methods, we're going to be limited to metabolism-based interactions only. With dynamic DDI simulations, we can unlock transporter interactions as well. With dynamic DDI simulations, what we're going to be doing is focusing on how competitive inhibition or time-dependent inhibition in induction act on the actual enzyme activities or metabolic or transporter rates. So with competitive inhibition, it's going to be, we have the ability to account for multiple inhibitors of a given enzyme and see how that affects the metabolic rates, multiple substrates for the given enzyme. With TDI and induction, we can account for multiple TDIs, multiple inducers, and in this case here, we're going to be focusing on how does the overall enzyme activity change with time. So for time-dependent inhibition and induction processes, we're going to be seeing how these perpetrators act on enzyme activities that will then be affecting metabolism or transport rates. Again, the heavy lifting with setting up dynamic DDI simulations are going to be in the development of the baseline models. Once we have those baseline models in place, then it's just going to be setting up whatever inhibition or induction parameters we have for the perpetrator. That's it. So let's go through one more activity here where we're going to be briefly setting up some perpetrator inputs for rifampicin, doing some simulations for auto induction, and then, if time allows, doing a quick prediction for an interaction with midazolam. Rifampicin is one of the models that we have as part of our standards database. The versions that we currently distribute were a lot of the work based on some nice publications from Roche that came out about 5 years ago. We are in the process of making some updates to that rifampicin model, which should be available soon. But we're going to be working off of the current model and talking about how we can leverage some of the data that we have to set up inhibition and induction parameters. So let's go ahead back into the software. I'm going to close out of the DDI module here and say yes to save my changes and update PK parameters. So if I wanted to come back with some new in vitro data, or I continue to refine and build up this risperidone PBPK model, I'll have a lot of those settings saved for me and can reuse downstream. But here, what I'd like to do is just open up an existing database that we have for rifampicin, one of the gold standards for evaluating 3A4 induction in particular. And so the inputs here, as mentioned, a lot of this has come from the Bannex et al. Publications from 2014 and 2015. So I'm not going to go through how we have set up the models here for the baseline models for rifampicin. It is a substrate for 3A4. It induces its own metabolism. It also serves as an inhibitor as well. So this is a relatively complicated model. And in addition to 3A4 metabolism in this current iteration of the model, there's also some clearance that is defined for non-CYP mechanisms, I think specifically esterases. And so there's an ability here to be able to run simulations to just see what the baseline results look like, a single dose. What do the simulations, how do the simulations compare to the observations look over the course of 24 hours? So this evaluation here of the prediction accuracy would be something that we do when we want to validate and verify our models. What I'd like to do is go back into that DDI module. So again, I'm skipping a lot of the heavy lifting of building up the baseline model first. But assume that we have that in place. If I go into the DDI module, remember the left side of this window here is where we see the current record, whether it's being treated as a victim or perpetrator. And for rifampicin, you can see we've already got populated here, if it's treated as the victim, the liver and gut FM percentages for 3A4, because we have set up the 3A4 enzyme in the enzyme table. But rifampicin is also a perpetrator. And so if I open up this Excel spreadsheet here where we have some perpetrator parameters, you can see there are some turnover rates which we enter into the main GastroPlus window. We can define degradation rates separately for the gut and or liver when we're using PBPK models. So that's been set up in the main GastroPlus program already. And what we want to do is just enter in here the inhibition and induction parameters for rifampicin. So if I select here that I want to add in some inhibition or induction values that I have generated in my lab, what I'm going to do first in this table is select the enzyme or transporter system. In this case, it's 3A4. And I'll enter first that I've got this total. So I've got to select here the drop down. It's in total in vitro competitive or reversible Ki value. And that value that is measured or reported is 18.5. And the units are micromolar. I also have some induction parameters here. So I'm going to add in another row. Also for 3A4. And in this case, I've got a total in vitro EC50. So I'm going to select here the total in vitro EC50. That value is measured at 0.8. And the units are micromolar. And then you'll notice in this in vitro experiment, there was also a reporting of the unbound fraction in the hepatocyte assay. And so what I want to do is make sure to enter that in. If I have a user defined or measured value, I'll select user here under the in vitro fraction unbound type. And then my value is 0.42. Now, this is going to cause for me a little bit of a mismatch like I discussed earlier. So now that I've got a measurement of the fraction unbound, the program will automatically convert that total EC50 into an unbound value. So what I should do for the competitive in vitro Ki here is use one of the calculation methods that the program provides. I could assume that my in vitro fraction unbound in this inhibition assay is equal to the fraction unbound in plasma, or I could use the Austin or Halifax methods here to estimate, to generate an estimate for the fraction unbound. And so if I select here the hepatocyte method from the Austin approach, assuming it's a similar assay here to the induction assay, I would generate the estimate of 0.856 here. So now I've set up, I also know that the protein content level from the in vitro experiments was 0.5, which matches up with my default values here. So I don't have to make any changes here. Only if the protein content was something other than 0.5 would I modify. So now I have some perpetrator inputs that have been set up and defined for rifampicin. And what I want to do here is run a simulation of rifampicin to predict and model any autoinduction or autoinhibition processes. So to do that, now that I have set up from the main record that my compound is a perpetrator, I'm also going to open up that same database. And I'm going to navigate to the same record here. And so now on the right side, I'm going to be treating rifampicin as the victim, but also during the course of these dynamic simulations that I'll run, also be utilizing the induction and inhibition parameters to simulate and predict autoinhibition, autoinduction. When I select to run a dynamic simulation, you'll notice that a lot of these labels are disabled and grayed out. Because with the dynamic simulations, remember, all I have to do here now is just select which inhibition or induction parameters I wish to use, set up the dosing information for the perpetrator, also set up the dosing information for the victim. So this is just going to be a simulation that I will run where rifampicin is dosed at 600 milligrams every 24 hours over the course of 7 days. You see here, when I select the dynamic simulation, that I have options to run either single simulations or population simulations. And I know in the day 2 course yesterday, Vieira spent time talking about the different population groups that are included in GastroPlus. And when I do these DDI predictions, I can set up these custom populations similar to what we would be doing in the full GastroPlus program. So any population simulations that are run from the main window can be reproduced here for DDI purposes. For purpose of time here, we'll just run a single simulation now. And you can see here the last step when doing the dynamic simulations, to run the baseline simulation first to establish the results, assuming no induction or inhibition mechanisms. We're just setting up what the baseline results would look like. This is assuming we're dosing with placebo and no interaction mechanisms are accounted for. And so you can see the progress down here in the lower left corner. And if I click on the show plot button, what you'll notice is that, okay, we're assuming no interactions right now, I'm dosing rifampicin 600 milligrams every 24 hours, and I have a constant peak and a constant trough. I'm not accounting for any autoinduction or autoinhibition. You see the squares, though, are some of the observed data from the real study collected on day 4 and day 7. And you can see that these concentrations do in fact drop over time. So here's my baseline results. And if I come back to the DDI window, I can choose here to now run the full simulation, which is where I will be accounting for the predictions. Oops, sorry, I forgot as one of the inputs here, the Emax value of 14.6 needs to also be entered in here when we're working with induction parameters. So my apologies, entering in that value of 14.6. And now if we run that full simulation, we're going to be accounting for any autoinhibition or autoinduction mechanisms for rifampicin. And I can see in this results window here, the last row is going to show me the ratios. So the ratios between the baseline set of results here, no interactions. The second row are the results assuming induction and inhibition mechanisms in place. And then the third row shows me the ratios between rows 1 and 2. And when I click on the show plot button this time, you'll see here now that I am accounting for, in this case, the dominant auto induction process. And I can see that my simulation results are dropping with time. So rifampicin just dosed by itself over the course of 7 days, I'm able to review and see, for example, how does the enzyme activity of 3A4 change over time as we're inducing metabolism, and how does that impact the systemic exposure? And you can see that there's a very nice match between the simulated and observed points going out across 7 days. So now, once we have done this kind of validation or verification, we would then be able to apply this rifampicin model to do predictions against other compounds. So I could open up either the standards database or open up any other databases for which I've created GastroPlus models of different drugs. And I could start running the simulations to do predictions now with rifampicin as an inducer and inhibitor. Seeing that I'm a few minutes over time here, this is a good place to stop. I just want to briefly summarize what we've done today in the course of 75 minutes or so is gone through some theory around static, steady state DDI predictions, some theory around dynamic mechanistic DDI simulations, and how we can start to leverage GastroPlus to be able to do the predictions at early stages to assess DDI liability. And then as we have more and more mature PBPK models, incorporate inhibition, induction, substrate information, do dynamic DDI simulations to track the changes of concentrations versus time, and have that be utilized to support not only internal decision making, but please remember, we can apply these GastroPlus DDI results to inform regulatory decisions as well.